We then just go through cytokinesis. I think you'll see we end up with two cells. They look like this. So, have we accomplished our goal of meiosis yet?
No.
We haven't, right? Roughly how much of our goal have we accomplished? About half of our goal, right? We've separated once, but we need to separate again. So what happens is it enters into another part of meiosis called meiosis 2. And so we can actually refer to this whole series of phases here as being in meiosis 1, or you could just call them prophase 1, metaphase 1, anaphase 1, telophase 1. Okay. So next, yes?
What is that thing on the side?
What thing on the side? I don't see anything. That was just a remnant chromosome from when I drew it differently earlier. Yeah, sorry about that. Okay, so what we're going to do now is we're going to go through meiosis 2. And I'm going to take these two cells, I'm going to draw them again down here. Sort of drawing them in, so they're not lined up, so you're more impressed when I draw them in metaphase. All right. So what will happen is this would be prophase 2. Then we're going to line these up in the center of the cell. In metaphase 2. This will be metaphase 2. And then we pull apart the sister chromatids from each other. So when we do that, we'll enter into anaphase 2. And so we'll separate these sister chromatids from each other. And our cells will kind of look like this as they're being pulled towards I'll leave this up while you guys are sketching, so you'll have this to sort of reference as you guys are sketching your own meiosis. And then we move into, I think you can predict, telophase 2. Where the cells are going to start pinching down, getting ready to divide. And I'm going to have one all open chromosome, and then the next cell will have an all open chromosome with that tip. They recombine from the other parent's chromosome. The other pair of cells will have an all filled in chromosome and one that has that open tip. They recombine from the other parents. And then we enter into cytokinesis, and bloop, we end up making 1, 2, 3, 4 cells. So are those cells haploid or diploid? How can you tell? They only have a single chromosome in them, right? So we went through and took a cell that was diploid, where it had a pair of chromosomes, and we copied them. We recombined the copies. We separated the pairs of sister chromatids. We then divided the cell. We took those two cells, we lined the pairs of sister chromatids up again. We separated the sister chromatids from each other, and then we divided the cell. So this set of cells here would be 4 haploid cells. And if these were coming from a female, it would be the egg. And if they were from the male, it would be the sperm. I'll talk a little, there's a little bit of a complexity to how females do meiosis that we'll talk about, not right now, but a little bit down the road. But this is the basics of meiosis. Do you see how in mitosis we took 1 cell and made 2 genetically identical cells? Here we take 1 cell and make 4 genetically unique cells. Do you see that? Every single one of those cells is totally different than the next. There's nothing identical about them. And if you do this across all 23 pairs of chromosomes and recombine, I don't know, I forget what the average number, 5, 7 per chromosome or something. You guys know?
Once per chromosome arm.
Once per, on average, once per chromosome arm?
I could be wrong.
Arm? I think it's more for the big chromosomes. I think there's more, because the number of recombinations is like related to the length of the chromosome. But when you think about that for all 23 pairs, you end up with virtually no chance you ever make an egg or sperm that's identical. They always are going to be mixtures of mom and dad's DNA and a random assemblage of those two. You combine them with another random assemblage from the other parent, you're going to be genetically similar to your brothers and sisters, but not genetically identical. Okay. Question?
So, real quick, just
Yeah.
on meiosis 1, there is no cytokinesis phase, right? It just is for phase 2.
There is cytokinesis. So this would be cytokinesis, because we did go from 1 cell to 2 cells.
Okay.
So you do have to have cytokinesis as well. And this would be cytokinesis occurring here. And then you guys get to do it for a 2n equals 10 cell. How lucky are you? That's right. We'll torture you until you understand this material. Okay, let's take a break, and then we'll attack.